Welcome back, AP Bio students. In this lesson, we will wrap up our study of meiosis. This is part 7 of Unit 4, and we're going to cover pages 208 to 212. Make sure you've read those pages before watching the lesson. So I want to highlight something that I mentioned in an earlier lesson. We talked about oogenesis and spermatogenesis, or collectively you could refer to these processes as gametogenesis. Remember, oogenesis begins with a primary oocyte in an ovary, and this cell undergoes meiosis to produce a haploid ovum, an egg, as well as some haploid polar bodies that are not functional, and they actually don't have much cytoplasm and they die. Spermatogenesis is when a primary spermatocyte, a diploid cell, undergoes meiosis to make 4 haploid functional sperm. And there are some differences, and there's a couple things that I can clarify with this image that maybe was not quite as obvious when we talked about this before. Prior to the start of egg making or sperm making, there are diploid cells in the ovaries and in the testes that divide by just regular mitosis. And in the ovary, these are called oogonia. This is an oogonium. It's a diploid cell in the ovary that divides by mitosis. But by the time a girl is born, that mitotic division has stopped. And once the ovaries have grown and developed, the mitosis part of this stops. And so there's a set number of primary oocytes at birth in each ovary. We talked about this before. And with the onset of puberty, 1 primary oocyte per month will undergo meiosis to make usually just 1 ovum per month. And that continues from puberty right until the point in the woman's life where both ovaries run out of primary oocytes. And once there are no more primary oocytes, there can be no more meiosis, and therefore no more eggs can be made, and that signals the onset of menopause. And so one of the big differences in gamete production between men and women is that in males, in male animals, the spermatogonia, this cell is called a spermatogonium, the diploid cell that makes up the testes. These cells continue to divide mitotically, making more and more primary spermatocytes. So there's a continuous resupplying of primary spermatocytes throughout life. And so even a man who's 90 years old still has a supply of primary spermatocytes, and they can still undergo meiosis and make sperm. The testes never run out of primary spermatocytes because of this continuous mitosis that's occurring, whereas the ovaries have a set number of primary oocytes once the mitosis comes to an end. And then just a little bit of clarification about polar bodies. The image that I used before was a little simplistic. This is a little more accurate. So in women, the primary oocyte, when it undergoes prophase 1, metaphase 1, anaphase 1, telophase 1, and it splits through cytokinesis, in actuality, it's a very unequal splitting of the cytoplasm. So almost all of the cytoplasm goes to this cell, this secondary oocyte, and very little cytoplasm goes to this cell. So we could actually call this a polar body instead of calling both of these secondary oocytes. It's really more correct to call this a polar body, and this is the secondary oocyte. And it has a lot of cytoplasm and plenty of mitochondria and so forth. So this cell is going to survive. It's going to continue through meiosis, prophase 2, metaphase 2, anaphase 2, telophase 2, and it's going to split to make two cells. But again, it's a very unequal distribution of cytoplasm. So almost all of the cytoplasm goes to this cell, which is going to be the functional ovum or the egg cell that may or may not get fertilized by a sperm. And actually, it's not until a sperm arrives at the cell that it actually completes the process of anaphase 2 and telophase 2 and actually finishes splitting. So it's kind of a little oddity of meiosis there. But the point I'm making is typically one ovum, one egg. And, before I said there would be three polar bodies, sometimes that's not the case. Now, if this polar body survives long enough to divide, polar bodies don't live very long. So if this polar body survives long enough, it will split into two polar bodies. And then you're going to get a third polar body when this secondary oocyte divides. So it's possible that there might be three polar bodies. But probably a more typical outcome would be the one egg and then just two polar bodies. Not that that's terribly important, but I just wanted to clarify that from earlier. Now in spermatogenesis, if we go back to the primary spermatocyte, this is the diploid cell that begins meiosis, prophase 1, metaphase 1, anaphase 1, telophase 1, and it splits. Both of these cells survive, and they have a pretty equal distribution of cytoplasm. So these would be the secondary spermatocytes. They both survive. They both continue through prophase 2, metaphase 2, anaphase 2, telophase 2. And when they divide, we get 4 haploid spermatids, and each one develops a flagellum and becomes a functional sperm. So typically all 4 do survive, as we previously discussed in men. Okay, so for the rest of, well, not the rest of the lesson, but for the next few minutes, I want to compare the process of mitosis with the process of meiosis. We've already done quite a bit of comparison in the previous lesson. I tried to, when I went through the phases, especially the first half of meiosis, I tried to show you with images how each phase of meiosis compares to the mitotic phases. But just to kind of wrap up, let's summarize the key differences between the two processes. So in mitosis, in prophase, the homologous chromosomes do not pair up. They don't act like pairs. They don't act like couples at the prom. In meiosis, that's a very big part of what happens. In prophase 1, you do have synapsis. Every chromosome that you inherited from your dad finds a partner that you inherited from your mom. So the red and the blue chromosomes, they come together as pairs, 23 pairs in fact in humans. And that coming together of homologous chromosomes is called synapsis. And it begins during prophase 1. Of course, it's really important because that's what allows crossing over to happen. It allows for that exchange of DNA between the homologous chromosomes. Mitosis has one division. Cytokinesis happens one time, and the one cell splits to make 2 identical daughter cells. But as you know, in meiosis, there are 2 divisions. The cell splits to make 2 cells, and then those cells both split again. There's a second division, so the 2 cells split to make 4 daughter cells. In mitosis, the 2 daughter cells are absolutely genetically identical. In meiosis, that is not the case. Each daughter cell is unique, has a different combination of genes. Mitosis is used throughout the body for growth, tissue repair, replacing lost cells, whereas we know that meiosis really only has one function, and that is to produce haploid cells that can be used for reproduction, either gametes or spores. If we're talking about a plant or a fungus, those reproductive cells will be spores. If we're talking about animals, those reproductive cells will be sperm or egg, gametes. Now, as we continue to compare and contrast the process of mitosis and meiosis, let's talk about three important things that only happen in meiosis and never, ever, ever happen in mitosis. The first is synapsis, the coming together of homologous pairs where every blue chromosome finds a red chromosome and they pair up. And they do that so that crossing over may occur, so that genes may be shuffled back and forth between blue and red chromosomes. Yes, I know they're not really blue and red. Chromosomes are not really color-coded, but that's a convenient way to picture it in your head. And that synapsis and crossing over happens in prophase 1 of meiosis. It never, ever happens in mitosis. And the second thing that happens only in meiosis, homologous pairs, or tetrads, as they're sometimes called, line up at the equator, the metaphase plate, double file, 2 by 2. Remember couples at the prom? Even if you make them form a line, they're still going to be side by side because they're all hugged up and they refuse to line up individually. They want to line up as couples. And that's what they do in metaphase 1 of meiosis. The chromosomes line up 2 by 2, double file. And they never do that in mitosis. When the chromosomes line up in the middle of mitotic metaphase, it's single file. They do not act like couples. And the third thing that happens in meiosis but never, ever, ever happens in mitosis is when you have sister chromatids still attached to each other and traveling to the same end of the cell. That never happens in mitosis, because in anaphase, when the chromosomes are pulled apart, one chromatid is pulled to one end and the other chromatid always goes the opposite way. But remember, in anaphase 1 of meiosis, the sister chromatids remain connected. They don't separate yet. They don't separate until anaphase 2. So in anaphase 1, both chromatids, both of the red chromatids go to one end of the cell and both of the blue chromatids go to the other end of the cell. If you just kind of picture a single pair of chromosomes, that's what happens. Both red chromatids stay together and go to one end, and both of the blue chromatids stay together and they go to the other end. So that's three things that only happen in meiosis and never ever happen in mitosis. So this is just a pictorial representation of what we just talked about. So, excuse me, so in prophase of mitosis, notice we have 4 chromosomes in this picture, 2 blue and 2 red. And this is a homologous pair, and this is a homologous pair, but they don't act like pairs. In mitosis, they don't pair up. They don't come together. There's no synapsis. There's no crossing over. But those same 4 chromosomes in prophase 1 of meiosis they do pair up. They form tetrads. They form couples. And they don't just pair up. They get really touchy-feely. And you have that overlapping between the non-sister, the red and the blue chromatids, so that they can do crossing over and they can swap genes. But that simply does not happen in mitotic prophase. There's no swapping of DNA back and forth between the chromosomes in mitosis. Because remember, in mitosis, we want both of these daughter cells to be absolutely genetically identical. We don't want to shuffle the genes. We want to make two exact copies of each other. But in meiosis, we're trying to make genetically diverse sperm and egg. And so we want to shuffle the genes back and forth. The second big difference you can see in mitotic metaphase of mitosis, the chromosomes always line up single file, whereas in metaphase 1 of meiosis, they line up double file. And then finally, at the end of mitosis, we have 2 daughter cells. Meiosis makes 4. And in mitosis, both daughter cells are genetically identical, and they both have the same number of chromosomes that we started with. In fact, if you look at this cell before before the DNA was replicated, here's what it looked like. And notice, if you look at the final product of mitosis, it's exactly the same. Same 4 chromosomes as the original cell. So mitosis does not change the chromosome number, and it does not shuffle the genes. So we get 2 genetically identical daughter cells, and they have the same number of chromosomes as the original cell. Meiosis produces 4 daughter cells. They are genetically unique, and they each have half the number of chromosomes that we started with. See? 1, 2, 3, 4 chromosomes in this cell, but down here, only 2. The number of chromosomes has been cut in half. Okay, one of the points that I made very early on in this unit is that sexual reproduction produces a tremendous amount of genetic diversity, genetic variation. A species that reproduces sexually is going to have lots of genetic diversity in the population. And so I want to get a little more specific now, and let's talk about 3 sources of that genetic variation. In other words, 3 things that happen with sexual reproduction that ensure that no 2 sperm cells are going to be alike, no 2 eggs are ever going to be alike, and therefore no 2 offspring will ever be exactly alike unless it's identical twins, and that's kind of a whole different lesson. But that's kind of the exception to the rule. Generally speaking, you do not look exactly like your siblings, even though you had the same mom and dad, perhaps. You do not have the exact same genes as your siblings, even though you and your brothers and sisters were produced by the same 2 parents. And there's 3 reasons for that. The first one is independent assortment of chromosomes. What in the world does that mean? Well, I think the easiest way to tell you is just to show you. So when the chromosomes line up in metaphase 1, they line up, double file, 2 by 2. And that's because the pairs of chromosomes came together and they paired up back in prophase 1. And so when the chromosomes line up in metaphase 1, they're still together. They're still paired up. And as they line up along the middle of the cell, it is completely random which side the blue chromosome lines up on versus the red. In other words, maybe the paternal chromosome is on the left and the maternal chromosome is on the right, but it could be the other way around. It could be the maternal chromosome is on the left and the paternal chromosome is on the right. It's random, and it's random for each pair. Now, this is a really simplistic example because they're only showing 4 chromosomes. They're only showing 2 pairs of chromosomes in the picture. But if this was a human cell, there would be 1 pair, a second pair, a third, fourth, fifth, 23 pairs lined up like this. And for each individual pair, it's completely random which side which side the blue chromosome is on versus which side the red chromosome is on. And that's important because remember, the blue chromosome and the red chromosome do not have exactly the same genes. All of the genes on the blue chromosome you inherited from your dad, and the ones on the red you inherited from your mom. So they're different genes, and which side they line up on is going to determine which sperm or which egg they end up in. So in this simplistic example, and this is really simplified here because we're only looking at 2 pairs of chromosomes, there's basically 2 ways they could line up. They could line up like this, where both of the blue chromosomes are on the left and both of the red ones are on the right. And that would produce 2 possible types of gametes, 2 possible combinations. But it's also equally possible that they line up like this, where 1 pair is blue-red and the other pair is red-blue. And that would produce 2 more possible combinations. So if you look at combination 1, it's entirely blue DNA. Combination 2 is completely 100% red DNA. Of course, even that is overly simplified, right? Because we know that this blue chromatid would really have a little bit of red on it, and the red would have a little bit of blue because of crossing over. So keep that in mind. I mean, this is not showing crossing over. This is just showing the results of independent assortment. But another possibility would be that the big chromosome has the blue copy, and the small chromosome has the red copy, or you could have the big chromosome red and the little chromosome blue. The point is, there's 4 possibilities. Now, there's a formula for calculating how many different combinations of paternal and maternal chromosomes can be created in the gametes. And the formula is 2 raised to the haploid power. Look at what this says. The formula for calculating the number of possible combinations of red and blue chromosomes, maternal and paternal chromosomes in the gametes due to independent assortment, due to the random nature in which the chromosomes line up. That formula is the number 2, always the number 2, but you put, as an exponent, you put the haploid number. So it's 2 to the n power. So this species, whatever kind of animal this is, it has 1, 2, 3, 4 chromosomes. The diploid number is 4. So that means the haploid number is 2. The haploid number is 2. So if we take the number 2 and we raise it to the 2 power, the second power, what is that? That's 2 times 2, which is 4. So there's 4 possible combinations of blue and red chromosomes that could end up in a sperm or an egg. Now, for humans, the haploid number for us humans is 23. So when you make sperm cells or egg cells and all of your chromosomes line up, 23 pairs of them, and each pair can line up blue, red, or red, blue. It's totally random. If you want to know how many different combinations of blue and red chromosomes can end up in the sperm or the eggs, it would be 2 to the 23rd power. That's 2 times 2 times 2 times 2 times 2 times 2, 23 times. Okay, and it's a big number. It's actually a little more than 8.4 million possible combinations. That doesn't even factor in crossing over. This is just independent assortment. It shows what a tremendous number of possible combinations are possible when sperm cells and egg cells are made. So let's do a sample problem. You could see something like this on an AP exam or maybe on the Unit 4 test. It says for a species with a diploid number of 8, we want to know how many different combinations of maternal and paternal chromosomes could be possible when gametes are made. I didn't phrase this as a question, but let's pretend that this is a question. How are we going to solve it? Well, the formula is 2 to the n power. The number 2, always the number 2, and then the exponent is simply the haploid number for the species. So if the diploid number is 8, that means the haploid number is 4. 2 to the 4th power is 2 times 2, which is 4, times 2 is 8, times 2 is 16. Again, I just took the number 2 and I raised it to the haploid power. The diploid number is 8, so the haploid number is 4. 2 times 2 times 2 times 2 is 16. All right, before I go to number 2 on the list, I almost forgot the extra credit phrase that pays. So here it is. 2 to the 23rd power is 8.4 million possible combinations. 2 to the 23rd power, that's the number 2 with the exponent of 23. 2 to the 23rd power equals 8.4 million possible combinations. That's your extra credit phrase that pays. Okay, we're talking about sources of genetic variation in sexual reproduction. First, we had independent assortment. It's totally random how the chromosomes line up, and therefore it's totally random whether a blue chromosome or a red chromosome ends up in a sperm or an egg. The second source of genetic variation, we've already talked extensively about this, so I won't elaborate much, but crossing over, as you know, is the shuffling, the physical exchange of DNA between non-sister chromatids. It occurs during prophase 1 of meiosis, and it causes the blue chromosome, the paternal DNA, to be mixed with some maternal genes. It causes the maternal chromatid to have some blue paternal DNA. And more importantly, what it does, it increases the number of genetic possibilities. It increases the number of different kinds of sperm and egg are possible by shuffling those maternal and paternal genes. And finally, the third source of genetic variation in sexual reproduction is random fertilization. And that simply says this. When fertilization occurs, and you have literally millions of sperm. Remember, there's 8.4 million possible sperm that can be produced just by independent assortment alone. And then you multiply that times all the the crossover possibilities, I mean, this number becomes almost an infinitely high, not really infinite, but it becomes a really, really big number of possible sperm. And they're swimming towards the egg. It's impossible to know which sperm is going to reach that egg. The sperm that gets there first is the winner, and that's the sperm that will fertilize the egg. But there's millions and millions and millions and millions of different possible sperm, and they each have a different combination of genes, and it's impossible to know which sperm will fertilize the egg. That's what we mean by random fertilization. So here's another little math problem that we could solve. It says a species of turtles has and I just made this up. I have no idea what the chromosome number of turtles is. You could look it up on Google, but I'm just going to make this up. A species of turtles has a diploid number of 6. Again, totally fictitious. Just go with me here. Based on independent assortment, how the chromosomes line up in meiosis, and random fertilization, let's factor both of those in. How many different genetic combinations are possible in the offspring? Wow. So first we have to calculate, well, when that male turtle makes sperm, how many different kinds of sperm can be made by independent assortment? And then when the mama turtle makes her eggs, how many different kinds of eggs can she make by independent assortment? And then we have to calculate all of the different possible combinations, because we don't know which sperm is going to fertilize which egg. That's random fertilization. So how do we do this? Well, first of all, the number of possible sperm that could be created by independent assortment, remember the formula? 2 to the n power. Well, the diploid number is 6. So what would be the haploid number? Half of 6, right? So it's 3. That's 2 to the 3rd power. You don't even need a calculator. It's just 2 times 2 times 2. 2 times 2 is 4, times 2 is 8. So that's 8 possible sperm that could be made by independent assortment. Likewise, when mama turtle makes her eggs, the number of possible eggs is going to be 2 to the n power. So diploid number is 6. That means the haploid number, the n number, is 3. 2 to the 3rd power, once again, is 8. So there's 8 possible sperm that could be made by independent assortment, 8 possible eggs that could be made by independent assortment, but we don't know which of these 8 sperm is going to fertilize which of these 8 eggs. It is completely random. So you have to account for all the possibilities. So that's 8 possible sperm multiplied times 8 possible eggs. That gives us 64 possible genetic combinations in the offspring. Now, think about this for humans, where our diploid number is 46. That would be 2 to the 23rd power. That's 8.4 million kinds of sperm that could fertilize 8.4 million kinds of eggs. So that's what, I don't know, 64 million. If I'm doing the math right in my head, I'm probably way off, but it's a bunch. It's a huge number. And remember, that doesn't even take into account all the possibilities that are created by crossing over, by that random shuffling of DNA between the blue and the red chromosomes. Doesn't even factor that in. This is only looking at independent assortment and random fertilization. We're only looking at 2 of our 3 sources of genetic variation. Crossing over is a big one, but we're only accounting for 1 and 3 in this problem. Guys, that brings us to the end of the study of meiosis. We're not quite done with Unit 4 because cell-to-cell communication, what are known as signal transduction pathways, how cells communicate with each other, in the body. That's another topic that's part of Unit 4, and we'll tackle that in a future lesson. But this is the end of cell division. This is the end of the study of mitosis and meiosis. I hope you've learned something. And remember the extra credit phrase that pays. Don't forget to email that to me. The first 10 receive extra credit. Guys, have a wonderful day, and I will see you soon.